Hello, and thank you for joining us this evening for our Big Day of Giving 2024 fundraiser. For those of you who are long-term supporters, we are so grateful to have you. And for those of you who are new to Headwater Science Institute, I should tell you a little bit about who we are. So since 2014, Headwaters programs have brought hands-on experiential learning to schools by providing extracurricular camps and various research learning opportunities to young learners. So support from you is going to, really means the world to us. It helps us continue to provide scholarship funding for our science programs. It helps us purchase materials for our school programs and continue to offer curriculum and activities free of charge to teachers. And guess what? I have exciting news. Tonight, for the next hour, all donations will be matched up to $500, and you can do so through www.bigdayofgiving.org/organization/headwaters. And go ahead, get those donations rolling in, help us reach our goals tonight, and we'll give you an update on where we are later in the evening. All right, so without further ado, I am going to introduce our guest speaker for the evening, Dr. Emily Meineke. She is a professor at UC Davis, studying insects and the plants that they eat. She uses very old specimens of plants and insects, along with field research in cities and vineyards, to assess what insects live, where they live, and how it's changing. Emily also teaches an art science fusion course at UC Davis. So let's welcome Dr. Emily Meineke. Welcome, Emily. How are you?
Thank you. Hi, everybody. Hi, Tara.
Great to have you. So, we are so excited that you are here today for our Big Day of Giving fundraiser. We are excited to learn about your research. I'm fascinated since I am a biologist myself and I studied insects, and I can't wait to hear more about what you do. So please, thank you for joining us and tell us a little bit about yourself and the work and your research.
Great. So I'm going to attempt to share my slides now. Tara, if you will just make sure that this happens properly.
Absolutely.
Okay, sounds good. Okay, are we seeing a slide with a mural on it and a title slide?
We see it.
Okay, excellent. Thank you. Okay, so I am a professor at UC Davis, like Tara said, and I'm about 4 years into my research. And so the way that my lab is and what I'm exploring is changing all the time. But I thought that this title sort of captured the things that I've been thinking about recently. The title slide actually contains a mural that was created in my art science fusion class at UC Davis. And all of the different pieces of the mural were created by undergraduate students, and it shows the inside of what's called a bee skep. So it's a human-made house for bees. And I'm not going to talk about this today, but I did want to use it for the title slide, just to show about some of the work that we do at UC Davis with art science fusion. And I think it could be cool at some point to collaborate with Headwaters on something like this. Okay, so I think it's a little bit of a black box sometimes, especially for people who attend college, me included, what a professor's job actually is. And so basically, the main part of my job is that I help young scientists become PhDs. And so the people who you're seeing in the The right side of the screen are two of my graduate students in those pink shirts, and they are doing an outreach event at picnic day. And a lot of my job is helping them design their science projects and find out what they're interested in. And then at the end, they become experts after about 4 or 5 years of studying with me and studying in the program at UC Davis. So most of my job is that, and coming up with my own research projects and pursuing them with the help of the graduate students and the undergraduate students in my lab. And then, of course, the other part of my job is teaching. And then a relatively smaller part of my job is outreach, which I'm going to talk a little bit about today, but it's also what I'm doing today. Um, so in my lab, we think a lot about climate change, and what you're seeing right here. Probably, I would ask you if we were in person, if you knew what this is. I'm sure a lot of you do. This is the big cherry blossom bloom in Japan, in Tokyo. And this was in 2018. And so I had a research trip that I was taking to Japan, and I was like, oh, I really wish that my research trip was going to dovetail with the big cherry bloom. And it turns out that it did, but only because the cherry blossom big event happened, um, the earliest it's ever happened before, in 2018. And that is because as the planet gets warmer, a lot of the things that plants do, especially plants that do things earlier in the spring, like put out flowers, those things are happening earlier and earlier. So if we look back in time, this is a diagram that shows when the cherry blossom bloom happened over about 12,000 years. And the data points are from diaries and periodicals. And so what you're seeing on the bottom axis there is year, so going back all the way to year 800. And then on the vertical axis, you're seeing March, April, May. And so if a flower dot is lower, that means that the cherry bloom happened earlier in that year. So if you go all the way to 2018, you can see that the dots are really low on the graph, which means that the bloom was super early. So you can see a lot of squiggles in that line, but in general, the cherry blossom bloom has been earlier recently, primarily because of climate change. So we know that this is happening for a lot of plants. What we know a lot less about is how this is affecting the interactions that plants have between plants and insects and plants and other animals. And these interactions are really what sort of runs the world and what makes our ecosystems do the things that they do. So think about things like whether animals are able to be out at the same time as plants and carry their seeds around the world and disperse them, and whether the microbes are there that need to be there in nectar for bees to eat as a supplemental food. So if the timing of when plants are doing the things they're doing is changing, and we have that recorded through various resources, we don't have similar resources that show us how the interactions with other organisms are changing. So one of the interactions that we know a lot about and that we think a lot about, especially in the agricultural part of North Carolina, I'm from North Carolina, from California, is interactions with pollinators. So it's really important that when the flowers are out, that the bees are also coming out to pollinate them. And so there are a lot of scientists that study whether with climate change, bees and plants are putting out resources at the same time and that they're both able to meet each other and make pollination happen. I'm not going to talk about that today because that is not what I study. What I think about is how climate change might be affecting interactions between plants and insect herbivores. So, insect herbivores are insects that are vegetarians, basically. So, they're munching on the plants all around us. Some of them are pests and they eat things in our gardens and they make them produce less food. The vast majority of insects that eat plants actually are not harmful, and they have relationships with plants that go back 400 million years. And so the reason why we have so many different kinds of plants on Earth and so many different kinds of insects is primarily because insects eat plants, plants make defenses against those insects. Insects develop new defenses, and that's how new species have been created over 400 million years. So most of the species on Earth are either plants or they're insects that eat plants, which is why I'm so interested in this type of interaction and how it's changing with climate change. And my daughter is 9 months old, and so I've been teaching her about this very early through the very hungry caterpillar, who's going to make another appearance later for anyone who might be a little bit younger in the audience, or I think all of us actually connect to the very hungry caterpillar. Okay, so this is a sawfly larvae, two sawfly larvae, and we might see the kind of munching that these caterpillars do. Basically, like when I go on a walk with my dog or something like that, I'll see, oh, a little bit of that plant is munched. Um, and sometimes that can have a small effect on how that plant functions. Oftentimes, it actually has a bigger effect than it looks like it's having. And so, even these tiny little sawfly larvae, if they eat about like 2% of the leaf, they reduce the growth of the actual tree by about 30%. So, if you remove just 2% of the leaf tissue, then you see 30% less growth. And so, the strength of these interactions and the effects they're having on the insects and the plants are actually bigger in nature than we expect them to be. And so that can have effects on things like decomposition, because when these caterpillars and these sawfly larvae and things like that eat plants, they're also pooping out what we call frass or caterpillar poop or beetle poop, whatever insect happens to be eating the plant. And that speeds up decomposition in the soil. And that can have all kinds of effects on nutrient cycling and other animals in the forest. So actually, these interactions look small, but they're a big deal and they're really old. So, given that we don't have data on insect herbivore plant interactions going back before the onset of climate change, before the onset of urbanization, all of the big effects that humans have had, how do we study these interactions? And so I started to get excited about this question when I was a postdoc at the Harvard University Herbaria. And I started thinking about how these pressed plant specimens, so herbarium specimens, might actually serve as a record of plant insect interactions going over time. So let me just explain a little bit about what an herbarium is, just in case you haven't spent time in one or been behind the scenes in a museum before. So herbarium specimens are most typically pressed plant specimens that people sort of clip off a plant, or they collect the whole plant and then they put it on acid-free paper. They press it in some sort of maybe a book. If it's a professional, maybe a professional plant press. They bring it back to a plant museum like this one called an herbarium. They put a label on it describing all kinds of things about the plant that they've collected. And then they put it in these folders that are arranged by what different plant species are, how related they are to one another. And then they sit there until a scientist like me wants to use them. Herbarium specimens also come in all of these other forms that you're seeing on your screen, including seeds and wood and things like that. But I'm not going to talk about that. So, interestingly, these pressed plant specimens, this is another example, they go back really far. So they go back into around 1700, and then more and more throughout the 1800s, people collected more of them. And the primary goal at first was to document life on Earth, to figure out how many species are there, what do they look like, can we create a museum that includes all of them? That that search still continues. We still have not described all of the species on Earth, but that search for plant specimens actually was one of the first natural sciences that women were allowed to participate in. And so it wasn't considered vulgar or improper for women to be botanists, at least as hobbyists. And so there were a lot of women who would go out into nature in the Victorian era with their petticoats and hike up mountains and collect these specimens. And so while a lot of the specimens that are accessioned in museums show that they're collected by men, often they were collected by their wives or their sisters or other women that they knew, which is another reason why these collections really interest me. So, in order to figure out if these specimens could serve as a record of plant insect interactions, I first had to figure out, well, are insects leaving traces behind of feeding on these specimens? You know, it's possible that people are trying to collect these really pristine specimens that don't have insect damage, and therefore we might just not see very much insect damage on them. But I thought, you know, insects are so common that it seems ridiculous that we might not see at least some. So I went back to specimens like these, which you can see are sort of relatively pristine. And I knew going in that people use specimens like these to study, for instance, how the timing of flowering has changed from the 1700s until the 1970s and beyond. So these two specimens are a little under 300 years apart. So you can tell something about when flowers were out by looking at the dates on each of them. And I thought maybe you could tell some ecological information about herbivory too. So what you're seeing here is caterpillar herbivory. And what I mean by herbivory is insect damage on plants, typically on leaves. And this is a specimen that was collected by Carl Linnaeus, who was the founder of binomial nomenclature, the naming system that we use to name species today, also in a very abundant collector of plants. And I was flipping through specimens when I was first thinking about this project, and I noticed that there are these different types of insect damage even on really old specimens. And so this sort of yellow, tannish, lightish area in the middle was probably created by a caterpillar who had really flimsy mouth parts and couldn't chew all the way through the leaf, but could chew partway through the leaf and then scraped off the leaf tissue. And so we do see abundant leaf damage on these specimens, was sort of the upshot of this investigation as I flipped through herbarium specimens. So not only can you see this sort of chewing done by caterpillars, but you can see more interesting and more rare types of insect damage or insect imprints on plants. And so on the left is an herbarium specimen of blueberry and this moon, three-fourths moon-shaped circular hole. And then on the right, you can see that megachilid bees, leaf cutting bees is what they're called. They actually create this kind of damage on leaves when they are carving out the parts of leaves that they use to then roll up and create their nests. So I just wanted to show a quick picture of this, of these leaf cutting bees and what they actually do with these three-fourth moon parts of the leaves. So they carve out their nests, which is really cool that you can see these interactions documented in these old specimens. Yeah, so there's the leaf sort of, the bee sort of sawing through that leaf there. Okay, so we can also see types of damage that we can trace back to individual insects. And so on the left is what we call a leaf mine that we can tell was made by the tiny little moth that is on the right side of the screen. And we can tell that that moth species made those tracks in the leaf because we can tell by the structure of where the poop is along the edge of the leaf mine is what we call them. And we can also tell by the shape of the mine. Okay, so I'm going to show you a little picture here of leaf mining, a little video of what that looks like. So typically you have an insect that lays an egg inside a leaf, and that leaf becomes both a home and a cafeteria to the larva that emerges from the egg. And so you might have seen these in nature, and often if you pull them open, especially during the spring or summer, you will find a live larva in there sort of eating through the leaf. Okay, so I thought it was really cool that we could see these different types of herbivory in the specimens, these really old specimens. And what I found out looking into herbarium specimens is there are about 400 million herbarium specimens that have been cataloged all over the world, collected since the 1700s, and accessioned at all of the herbaria you see here on your screen. and all the green dots and all the stars you see are all different herbaria. So I had to figure out where to start in my investigations of looking back in time at insect herbivory in these specimens. So what I decided to do, since I was at the Harvard University herbaria, is to first focus there. And in the northeastern US, where I know some of you who are on this call are actually located, it turns out that winter is warming quite a lot. And so what I wanted to study first was this prediction that herbivory in New England has increased over the past century because of warmer winter temperatures. And so what has typically happened in the past in New England is that it gets really cold and insects don't like it that cold. Some insects survive, a lot of them die over the winter. And so I thought, well, if winters are getting milder, maybe that means that we will see insect herbivory decline over time as the insects die less. Okay, so I chose to focus on 4 species that are both common in New England forests, but also have important functions. So it's these 3 species. Desmodium canadense serves as a host plant for a lot of rare butterflies. Vaccinium is a group of blueberries that lots of animals eat. And Quercus bicolor and Caria ovata, white oak and hickory are serving as important food plants and canopy species for the forest. And then I went to a bunch of herbaria, including the University of Vermont, that's pictured right here. And I looked at all of these different holes in leaves and I quantified how many of the specimens had these holes in their leaves created by caterpillars and beetles and various other things that have mandibles and chew through leaves. And then that allowed me to figure out when a specimen was collected and then how much insect damage was there, with the prediction that as winters have gotten milder over time, maybe insect herbivory has gone up because the number of insects that survive winter has gone up. And primarily, we did focus on types of herbivory that are most likely made by these very hungry caterpillars. Okay, so I'm just going to go directly to what we found. So what you're looking at here is a graph of herbarium specimens. Each dot is an herbarium specimens of different plant species. On the horizontal axis is year a specimen was collected. And then the probability of a specimen having insect damage of some kind, so some chewing on it, is what's on the y-axis. And so specimens that had insect damage are up at the top with a 1, specimens that didn't are at the bottom with a 0. So what this showed us was that no matter which species you look at, even though all these species are not really related to one another, nor do they support similar insect communities, that across all of them, insect herbivory has increased about 12% over this time period from 1890 to 2010. And similar pattern if you look at a graph of temperature and probability of herbivory. So it turns out that herbivory goes up over time, and that as winter temperatures get warmer, herbivory also goes up. And so what seems to be happening is exactly what we predicted, that insects are doing better as it gets warmer. So just to show you sort of a very hungry caterpillar description of this, historically in New England, it's been chilly in blue. A lot of the caterpillars die out. Now it's a bit warmer in New England and a lot of caterpillars do really well. That's what we think is happening here. We also think another thing that might be happening that we haven't tested yet is that as it gets warmer in New England, new species that were historically further south are actually moving further north. So the moths are able to disperse. As the winter gets warmer, the caterpillars that they have are able to survive better. And so not only might we be getting more of these hungry caterpillars, but if you imagine, we might be getting different colors and shapes of these hungry caterpillars that are new to the area. So this narrative is kind of in contrast to what we know about insects, right? I'm not sure if you all know, but there is a call for people to be aware of the fact that a lot of insect species are in decline. So what this figure is showing us is it's a big study that was brought together a bunch of other studies, and it was published in a big journal. And the upshot of it is that those brown boxes tell us that insect abundance is decreasing about 9% per decade overall. And a lot of this data was from Germany, but it applies to a lot of the globe. So why might we be seeing more insect damage on leaves while insects decline themselves? And what we think might be happening is that while most insects are in decline, they're either blinking out completely or there are a lot fewer of them than there used to be. We think there are certain very hungry species that are doing really well with climate change and are making most of the damage that we're seeing. So we don't think that this pattern is due to like a bunch of species doing really well as the winters get milder. We think there are just a few species that are probably benefiting from climate change that are creating this seemingly discordant pattern between insects and the plants that they eat. Okay, so we're pretty convinced about the utility of herbarium specimens as records of plant-insect interactions. And what I want to talk about now is herbarium specimens as a way to communicate about climate change to a broad public audience through an art science fusion project that I participated in. And so this is a description of an exhibition that will be up, I think, until May at the Harvard Museum of Natural History. And it's called In Search of Thoreau's Flowers, an Exploration of Change and Loss. And I hope those of you who are at the Harvard University herbaria or nearby will go and visit. Okay, so I worked on this in collaboration with Marcia Gordon, Robin Vuknik, Leah Sobsey, and Chuck Davis. And Leah and Robin are artists. Marcia is a historian, and Chuck is a botanist at Harvard. Okay, so I first want to show you a little video of what part of the exhibit looks like. So these are two little girls who are sitting next to an interactive part of the exhibit in which plants that used to be in Thoreau's woods are blooming beside them. These same plants are not in Thoreau's woods as abundantly anymore, or they've gone extinct, and we were able to bring them back to life using parts of herbarium specimens. So I'm going to talk about that. This is the other part of the exhibit. And so what you're seeing here is work by Leah Sobsey. On the left are portraits of plants that have either gone extinct or are doing really poorly in the region now. And on the right are a bunch of herbarium specimens of different plant species. And the ones that are in blue are the ones that are not doing very well with climate change. Okay, so I want to take you back to the impetus for this exhibit and the inspiration, which was Henry David Thoreau, who was a transcendentalist biologist and botanist in the 1800s in New England. And so you may have heard of Thoreau's woods. He is known for creating detailed journals where he wrote down, just like people in Japan wrote down about the bloom of the cherry blossoms. He wrote down when various plants in Walden Pond in what's now called Thoreau's woods, when they flowered. And so this is a quote from Thoreau. I soon found myself observing when plants first blossomed and leafed, and I followed it up early and late, far and near, several years in succession, running to different sides of town and into the neighboring towns, often between 20 and 30 miles a day. So in Thoreau's journals are these records of when different plant species used to flower. And as he collected these data on these plants, he also collected specimens of these plants. And these are actually specimens that are now digitally available and became digitally available while I was at the Harvard University herbaria. And I thought, oh, we need to do something with these specimens that just became available in digital form as an art installation. And so I've showed this before. Basically, what it shows you is that herbarium specimens can also tell us something about when plants used to flower versus when they flower now. So people went back to the same areas where Thoreau collected his really thorough data on when plants first blossomed. And in more recent years, starting in I think 2006, they recreated those data. So people looked at when the plants that Thoreau looked at, their same species, when they blossomed more recently, and found that some plants didn't change at all. The vast majority now bloom a lot earlier than they did during Thoreau's time, and some of them are actually delayed. And it turns out that the plants that are able to keep up with climate change, the ones that are able to keep up with warming and therefore bloom earlier in warmer years, they are also the lineages, the groups of plants that are doing really well with climate change. And so it seems like there's some benefit to, if it gets really warm, you're able to put out your flowers and sort of track the local climate. And that is the reason why some plants seem to be doing really poorly now in Thoreau's woods, is that they just can't keep up with climate change. Okay, so the other, this is a little bit of whiplash, but the other inspiration for this exhibit was Anna Atkins. So she was around in the 17 and 1800s, and she was an English botanist and photographer. And she's the first person to publish a book illustrated with photographic images. And she also is one of the first people to use what's called cyanotyping. And so Sir John Herschel was the first one to create cyanotypes, which are photographs that are created using UV light and that create these like blue style photographs. And Anna Atkins applied this process to seaweed and made these photograms that were able to capture the physical structure of seaweeds when there's really no other way to preserve them. And so we don't have a lot of sea plants actually in herbaria, because as soon as you bring them up from the ocean, they tend to sort of fall apart right away. So Anna Atkins figured out a different way to create herbarium specimens of these species. So we wanted to also highlight the work that she did in our exhibit. And so these two people together kind of came together to result in this exhibit that the four of us put together. And we're a really interdisciplinary team that used the digitized collection of Henry David Thoreau's pressed plant specimens to reflect on environmental change. So these are some images of Henry David Thoreau's original specimens that he collected in the 1800s. And what we did was we went through all of the data that we had from Thoreau and from others about when plants flowered and how well they were doing in Thoreau's woods. And so this is just a little snippet of the dizzyingly abundant data that we had to work with. And we used these data to figure out which plants were in decline. And this is one example of a plant that's in decline that Leah, one of our collaborators, turned into a cyanotype. So we basically wanted to reflect on the fact that these plants were in decline by shading them in blue. And so these are some more examples of images that Leah created of these plants that are not doing very well now in Thoreau's woods. And again, she turned it into this installation that highlights the plants that are doing well versus the plants that are not, that are shaded in blue. So this is quite a big wall. And she also did these portraits that are Victorian era portraits, so the same era that Thoreau was around. And she wanted to honor these plants that we have caused to either go extinct or to not thrive as a result of climate change. So the other aspect of these that I think is really important is that people can see their reflections in them, which is a part of the message that Leah wanted to convey. So there's also Thoreau's original text. So he was good at a lot of things. Handwriting was not one of them. And so she put some of his original writings in the back of these cyanotypes. So just showing you some other images from the exhibit here, and we find that people really like to touch the screens. This is an installation on the floor. I don't know if you can hear it, but we collected sounds from Walden Pond, from Thoreau's Woods, and overlaid them as an audio installation in the exhibit so you can hear what it sounds like there. And Robin Butchnick, the other person who helped with this exhibit, sort of made the plants come alive on the floor through use of the herbarium specimens and projections. Finally, I think the best part of creating this exhibit has been how excited kids are to see the plants behind them blooming, and also the floor exhibit, and how interactive it has been for them. And I want to sort of end by showing you this slide of where you can find more about the exhibit. So it's at the Harvard University Museum of Natural Science, and we actually have a website where you can check out more if you're interested. So at this point, I'm going to stop sharing my screen, and I'll be happy to take any questions that you have.
Wonderful. Thank you so much. That was just really amazing. Your research in urban herbivory is just fascinating, and I really love the way that you tied in historic preservation and art into your work. But less talking from me. Let's hear from some of our audience members. So I'm going to just bring some folks in. Hi, Jen.
Hi. So we have a couple of questions in the chat. First is, generally, which plant families seem to be the most able to adapt to climate change?
I know more about the ones that aren't, so I'll say the ones that aren't.
That's
Lilies
fine too,
and
because we need to know that.
violets
That's information.
and orchids and things like that are not doing quite as well as some of the other plants. Yep.
Okay, and then we had another one. But first of all, the two people that are asking these questions are also part of our board, so they are watching, and they're so excited to hear you talk. So it's a long one, but certain insects are benefiting while others are declining with climate change. Can this also be linked partially to the use of pesticides? That's a very good question, because that's a hot topic, right? Especially with our students, is the use of pesticides and how that influences our environment. So the use of pesticides killing insects and or genetically modifying plants, resulting in reduction of a food source. So very awesome question. Thank you, Donna.
Yeah, that's a wonderful question. So it's interesting. Like, one of the things we think about a lot in my lab is the fact that we talk about climate change a lot, but actually the main driver of insect decline, and all of these studies that have looked at it, is the loss of their food plants that results from habitat destruction. So things like urban development and agricultural development, getting rid of plants that insects and the food webs above them rely on. So and certainly pesticides are also contributing. And so if you look into the declines of butterflies in this area in the Central Valley of California, the main driver is the introduction of mass use of pesticides. I am unaware of any evidence, actually, that genetically modified plants are playing a role in insect decline. It seems like in a lot of cases, actually, genetic modification is just changing like a tiny thing about a plant that makes it more able to withstand insect damage and provide mass amounts of food. And so while monocultures are certainly not like a thing that's good for insects at a large scale, I think genetically modified food is probably something that has more of a complex effect.
So we have a lot of students that pay attention. Can you explain what the difference, like what a monoculture is versus?
Oh, yeah, sure.
Yeah, we just have a lot of kids that do watch, and this is something that we send them.
Oh, I'd love to talk about it. Okay, so a monoculture is basically like, imagine a cornfield. So it's when you take one crop, usually one that we feed to animals that we eat or we feed to humans, and you have it in a really big field and you plant all one thing. And the thing about that is, number one, that we often have to spray those places with a lot of pesticides, because if you don't, you get a lot of pests that build up because they have tons and tons and tons of food. Whereas in the forest, where you have lots of different kinds of plant species that support different insect species, a lot of times those insects will compete with one another and sort of keep each other in check. The same thing is true if you have farming that's not a monoculture and you include lots of different plant species. Often that ends up creating more diverse insect communities than if you have just one crop planted.
Awesome. So I'm going to ask another question before we go to more, because I used to teach AP environmental science. So going along with what you're talking about, we talked about integrated pest management. Do you know anything about that? Like Biggest Little Farm, beautiful documentary. I don't know if you know anything about integrated pest management, or if you can talk about it a little bit.
Yeah, so I teach a class at UC Davis called Biological Control. It's one of the things in the integrated pest management toolbox. And so integrated pest management is basically like a common sense way to deal with insect pests, insects that are, typically eating our food. And so you go through these steps of figuring out, like, what insects do I have? How many of them can I tolerate before I need to have some sort of control measure, meaning like spraying them with a pesticide or removing them individually if you have just like a little small area in your garden. And it's a method of not just like spraying pesticides on a schedule, so every 2 weeks going out and spraying them. It's instead like really getting to know your ecosystem and using all of the information you have to make fully informed decisions. That's basically what integrated pest management is.
It's ladybugs. We love ladybugs. They're so cute, and they
Oh
eat
yeah,
everything.
and they're vicious.
They are
Not to
vicious, but
us,
they're so cute. They
but
land on you and it's good luck, but they'll also eat everything.
they do. They eat all the pests.
So I'm going to bring in Jeff Higginbotham. So Jeff is the lead secondary science context expert here in Brevard Public Schools here in Florida. I've known him a very long time. He's highly intelligent, amazing, loves microbiology, but he has a couple of questions that he wants to ask you. So I'm going to go ahead and let Jeff take it away.
Hi there. So thank
Hi,
you for
Jeff.
having me on. So my primary work is with middle and high school students. And one of the things that I do is I help run our secondary science fairs. So what advice would you give these student researchers trying to compete in these science fairs? And how would they get into the type of research that you are currently doing?
Yeah, interesting. So I think in science, you can have lots of different goals, right? And I think that it's fine to win a science fair. Like, that's really exciting. What I think is more exciting is if you can find something that you're really interested in to dig into. And so I think the bigger prize is if you come out with a project that you're excited about and proud of. And even if it doesn't win a ribbon, you know, you've really won. So I would suggest, and this is what I tell to my graduate students too, is that really spending the time to figure out what makes you light up inside is the biggest first step. So that would be my advice, is to focus on that.
Thank you.
Awesome. Yeah, Jeff does. So Brevard County is really big with science fairs. So thank you for giving those kids that answer, because we try and tell them all the time, but hearing it from a scientist is going to make such a bigger impact. Tell them till we're blue in the face. So hearing it from someone like you is amazing. So thank you so much, Jeff.
Thank you.
Do you have any other questions or? You good?
No, that's the big one.
That's the big one. That's the big one. We always want to know that. So thank you, Jeff. We had a really cool question in the chat. Do you think as climate change alters ecosystems and some become cannot ever say this, homogenous, do you think we will see altered herbivory, God, my brain, herbivory patterns and even potentially species change?
Yeah, so this is a really good question. So I think that ecosystems are in some ways becoming more homogeneous. Most of that, instead of being because of climate change, is about the introduction of invasive species. So you have invasive plants that are making their way around the globe. And so what that ends up doing is making all of the flora, the plants around the globe, look a little bit more similar. Of course, in a lot of places, you're still getting plants that are specifically from that place. Florida still looks like Florida. California still looks like California to some extent, with the exception of cities, right? So we have a lot of species that tend to live really well in cities all over the world. So there are pigeons in a
Oh,
lot of cities.
yes.
There are rats.
Every single place I've ever been has had a pigeon. That is correct.
Dandelions, yeah, four-leaf clovers, three-leaf clovers, you know, so I do think that that is going to continue to happen, that our cities are going to continue to get more and more similar as because the reason why things move around to different places is because we're carrying them. So either on cargo ships or airplanes or, any mode of transport that humans use, there are going to be seeds and various things that travel with them. And so that I think is more of a source of homogeneity than climate change will be.
So it's really interesting that you say that. So I live in Florida and we have crazy stuff. So everything that gets brought here on like cargo ships or luggage or anything like that thrives here. And I think, there's so much research going on trying to figure out where the limits are for these invasive species. But it's really interesting you say that because we have where I'm at over by the Space Center, curly-tailed lizards have completely taken over everything. And they're from Cuba originally. So it's pretty far south where we are. And it's a tropical climate versus a temperate climate. So it's really interesting for you to say that because, I think things can just adapt, right? Like that's all part of biology and how life goes. Life will find a way. That famous quote by Jeff Goldblum. That's one of my favorite things ever. But yeah, it's pretty insane. So maybe it's not climate change. It's just life's just going to find a way.
In a lot of cases, I think that's true. Although, if you introduce the curly-tailed lizard to Toronto, it's going to be like, no, thank you. So
that would be disastrous. Maybe I'll ship them all up there because they are abundant. All right. So next, I'm going to bring in Lynn. Lynn is an ecotourism superstar here on the west coast of Florida. She has a couple of questions. She put one in the chat, but I'm going to let you ask it firsthand, Lynn.
Hi, Emily. Thank you so much for being here tonight. And actually, I'm really glad that they just brought up the last question concerning climate change because in Florida, as you know, we were just talking about all the different animals that are invasive, but climate change will start to cause a problem with losing plant species when the oceans rise. The Miami blue butterfly is only found in the Keys, and the Keys are going to be underwater in a little bit. How do you then track? You have plants from the herbivarium that you can go back to, but then a species is gone. And maybe the Miami blue adapts, but what happens when you actually lose a species because of either exotic took over or because you had climate change wipe it out?
Yeah, so my friend Erica Henry did that research and we were in graduate school at the same time. So I remember learning about the Miami blue, and it is quite a difficult example, right? Where you have a species that's native to a place and only lives there. It's its last habitat. It's arguably a remnant of what might be a larger habitat. And eventually it's going to go underwater. And I think the sad truth is that it's funny, even though Erica knew that, she still studied it and still wanted to understand how we could preserve it for as long as we can, because she cares that much about nature. The sad truth is that it will go extinct unless someone creates a captive colony, so a colony of these, and then tries to reintroduce them or introduce them into a different area that's not going to go underwater, then we'll lose it forever. And that happens all the time. Species are going extinct all the time, and unfortunately, evolution doesn't work fast enough for us to replace species as they're being lost. Everything that we're doing is a lot quicker than how nature in the past has done things, because we're such an impactful species. So we've had warming events in the past, for instance, but they've never happened as quickly as they're happening right now. So unfortunately, lycanthropes woods with the plants, they'll just blink out one day. And that's why we have to care about climate change and try and reverse course at a global scale.
Yeah, we're experiencing a lot of that in Florida. And
I know you are. Mm-hmm.
Also, you talked about the winters getting warmer up north. I'm from Boston, so been there, done that. Florida's seeing an extreme heat. Our temperatures are higher. Our water temperatures are just getting kind of crazy, insane. And I know that some species, you were talking about how flowers bloom faster. Well, we have the tussock moth that, it waits for certain temperatures to come out. Are you seeing a trend with, not only are the plants acting differently, but animals are maturing, dragonflies are coming out of the water sooner because of this? I mean, this isn't just the plants causing it. This is a lot, isn't it?
Yeah, totally. So spring is a race.
Yes.
Right? It's like, it's warm. You're not going to freeze anymore. In Florida, you were probably never going to freeze, but
not a little bit.
In a lot of the worlds, you're not going to freeze anymore. Okay, let's get out there and do our thing. Let's put out our leaves. Let's put out our flowers. Let's mature into adults and fly around. So plants are definitely not the only things that are taking cues from the temperature. And insects and the plants that they eat might actually be responding to exactly the same things that are happening in the environment in the spring and deciding when to come out using the same, what we would call, cues. The challenge is that we don't know what the vast majority of insects and plants, what they use specifically to decide when to do their thing, what it is. So usually it's some combination of of the day length and the night length and temperature. But a lot of plants require for it to get like a certain level. It's called chilling down in the winter. They need to get really cold before they can do their thing in the spring.
Right.
Similarly, a lot of insects need to what we call diapause, so they need to get in a really cold state before they can do their thing in the spring. So the point is that insects and plants can be closely associated with one another in time, but use pretty different cues. And so as the planet warms up, if an insect is using minimum temperature and a plant is using mean temperature to decide how fast to develop, they might become decoupled. Similar to if a plant is primarily using day length, but an insect is using temperature, they might become decoupled. And so people are studying that a lot now. And it seems like for bees, at least, that there is some things will get out of sync, they'll get in sync again because there's enough pressure for them to figure out a way to like be together in time. But with herbivores, it's a little bit different, and we're really not sure exactly what's going on. So
there you go. There's the next science fair project, right?
totally. Yeah. That'd be a great science fair project.
I just
Thank you so much, Emily.
Thank you, Lynn. Yeah.
Lynn, you're fantastic. Yes. Our kids need to be able to do this stuff. And you know what's really fantastic is that Headwaters can do that with the kids. Like we can get them involved. In fact, that's what this whole thing is about, right? We have Big Day of Giving today. Every single donation that you make goes back to our students. So we have such an amazing opportunity today to donate and get our kids involved. We have this link up. We've had it up this whole time. In fact, we even have an amazing science camp this summer. So if you want to get your kids involved in our summer programs in the Truckee Tahoe area, we also have an online program that is for our research experience. We have all of our information on our website. So sorry, I had to do a little bit of plugging, but we also have some questions that are still running. Emily, you're blowing up our questions. So we normally don't get this many questions. So you're doing an awesome job. Are there other long-running data sets that could be of use to scientists that are studying climate impacts? Thinking about outreach, how can scientists reach out to communities that might have useful data? So there's a lot of citizen science projects going on. And also, you know, talking about those long running data sets, right? Like kids can leverage that in science fair and also just as a community, like a citizen scientist trying to do research.
Yeah, we're always trying to find new sources of data. And I'm sure there are so many that are just latent and in communities and not being used by scientists yet. And so it'd be so cool to see a kid make use of those, especially in collaboration with a scientist. And yes, people are collecting contemporary data sets with citizen science, which is also useful because it covers a really large spatial range,
Yeah, it's very
which tells us what's happening all over the world.
Mm-hmm.
So honestly, our long-term data sets that we know a lot about come from you know, a few eccentric individuals who got really interested in one thing and decided without any praise or input from the rest of the world, they were going to keep track. So there was a guy in Europe that, I think he was in Denmark, who collected bat poop for 50 years. And people are now using that to pull the DNA from the bat poop and see how what bats eat has changed over the past 50 years.
Well,
There's
they're an evening pollinator, and people don't really think of bats that way. And like bees, bats are your nighttime, your nocturnal pollinators. So that's a really awesome thing to share.
I can't say enough about bats. I love them. And I love that this person just decided to go out with 5-gallon buckets and collect bat poop and make a room with it. Like what
That's
a true scientist.
That's how you become Batman. Like you have to collect all the guano. Like you just go out there.
It's a true Batman. Yeah,
That's
and something similar. There was a dude in Colorado who did something similar where he like looked at weather conditions and the amount of snow that was in a particular area every single day for a really long time. And now people are using those data to see how it compares to the amount of snow that's there now. And I could keep going. I mean, there are all these individual people who are just like, I'm going to do something that I think is pretty cool. And now scientists are using that information because it covers a time span that we really don't have a lot of information on.
Listen, science nerd, we rule the world, guys. Like get out there, collect your data, make some observations. Like you don't know if you're going to be the next person that's big. So we have one more we have 2 more panelists. So we have Natalie, who is joining us from the Reno area. And then we have our executive director that's going to tell you guys a little bit about why you should help us out here for Big Day of Giving and the amazing benefits that your kind donations are going to provide. So I'm going to pull Natalie in. Hi, Natalie.
Hi, Emily. Thank you so much for speaking about your research and the art installation, and then Headwater for hosting this wonderful panel. I did have two questions. The first one is a science-oriented question, and then I had a question about the art installation as well. So for the science question, could you maybe explain the insect sort of chain reaction? I know you've spent a lot of time talking about the herbivorean side of the insects, but could you also speak to the insects who aren't herbivorians that are also affected by the hungry caterpillar, if you will? Like what happens to the bee population or other insects that are also reliant on these plants?
Yeah, so often, almost always, insect herbivores have predators that are eating them on a regular basis. So birds are a big one. Birds love caterpillars and beetles and things. Another one is insect predators. So we were just talking about ladybugs, all manner of beetles and various other things. Then we have parasitoids, which are these tiny, often beautiful metallic wasps that lay an egg inside various insects and then develop as larvae inside them and sort of eat them from the inside out. It's this really cool,
Wow.
like zombie thing. I would encourage you to look up videos of parasitoids if it seems interesting to you. So all of this diversity of creatures really depends on the herbivores that are eating the plants. And so when something happens to the herbivores, then it has knock-on effects on both, vertebrates like birds, but also invertebrates, which make up the majority of the species that are eating the caterpillars. And so one thing that we do know, we don't know that much. One thing that we do know is that the predators and parasitoids are much, much more sensitive to climate change than the herbivores or the plants are. So they tend to drop off before anybody else. And we don't know exactly why. So
yeah, no, I really appreciate you explaining the whole chain reaction so we can get a circumspective view of really the ecosystem. And then I really enjoyed your presentation on the art installation. I noticed a bit of irony that you sort of had the viewer, man, if you will, also interact directly with the installation, sort of like an analogy to climate change. Was that accidental, or was that implied that you wanted an interactive art installation where man is literally acting in conjunction with the art, similarly to how us man have sort of, I guess you could say, been the catalyst of climate change? Was that intentional, or was that merely an afterthought?
that's interesting. I hadn't really thought of it as a mirroring. What I really wanted was, I think that today we find things so much more exciting. Number one, if they're digital, and number two, if we can feel like we're immersed, And I think that a lot of what happens with media and climate change is that it makes people want to turn away. So this might have something to do with me being a climate change researcher, but when the New York Times publishes another article on climate change, I'm just like, I can't engage with it. I can't read it. Like, I know it's bad. And so I really wanted for us to create something that felt immersive and beautiful enough to encourage participation in learning, because I felt so disengaged from things that were trying to teach me about climate change. Yeah, does that answer your question?
Yeah, I mean, I think it's always an exploration of man and nature, right? I guess my follow-up question to that was, walking away from the installation, was there an intentional approach outside of outreach to sort of bring the viewer to go research other ways other than pesticides and pest management, to sort of lean into, I guess you could say, the race of spring and herbivorian interactions? Or is that something that's sort of unknown at this moment with respect to your research?
We, at the end of the exhibit, I'm not sure if I'm answering your question, but at the end of the exhibit, we did provide some information on how people could encourage more biodiversity, like in their yards, by planting more native flowers and just being a bit more aware of the pesticides that
Right.
there's a class of pesticides that sort of shows up in plants, for instance, from lows and stuff, and gets into the pollen and the nectar. And it's
Right.
not always really well advertised which plants those are when you go to buy them, which is kind of a bummer. So being aware of those little things can really go a long way to promoting insects and their function. And so we did leave viewers with some information about that at the end of the exhibit. We didn't talk a ton about herbivores in that exhibit just because flowering and decline of the plants has been a bigger story in Thoreau's Woods, and we really don't know that much about what's happening with the herbivores, but we did highlight that the specimens are a source of data on those interactions. Yeah.
Awesome. And then just briefly, is there anything you want us as basic civilians to sort of share? Like, how can we carry the torch for your outreach messaging if we didn't get to see the exhibit, or just in general?
Yeah, I mean, I think the things I just said are good ones. Like just being very aware of what you plant in your yard, like
Mhm.
every plant that you plant with flowers, because it's providing pollen and nectar. It's supporting not only bees, but all kinds of other things that rely on pollen and nectar, including predators and parasitoids that require those alternate food sources to be able to eat herbivores. So I think just doing the good work of being a steward of your own yard is like, it just goes a really long way.
Yeah, thank
Yeah.
you so much for answering all my questions. I really appreciate it.
Yeah,
Excellent.
thank you for your questions.
Thank you.
Thank you so much. Well, wow, what an hour. That was really just so insightful and eye-opening. And yeah, about the pesticides, just totally fascinating here in Reno, Nevada, or in Nevada in general. AB 162 was signed into law by Governor Lombardo, so that hopefully bans a lot of those pesticides that are showing up in plants that you're getting in, your regular box hardware stores. But I hope to see things like that, getting passed across the United States. But thank you again for being here. I just want to throw our executive director, Meg Seifert, on to just give a little, plug for Headwaters. And here she is.
Hi, Emily. Actually, Meg Seifert. Thank you so much for joining us tonight. We try to inspire, students in science and raise money to do that. I was just curious how, what inspired you to be, a woman in science, to push through that? Like, when did you decide? Because I think that's really important, for students to hear from people that we're working with,
Yeah, I will say I don't know exactly. I think in college, I think that there's this expectation that there's going to be this light bulb moment, right? Where you'd be like, I was destined to be a scientist, or I was destined to be a mathematician. And then when you get there, you're like, well, that's not how that works. You just kind of keep rolling down roads that seem interesting in the moment. And so the way it worked for me is that I kind of got to college and was like, I'm tired of being in a classroom, kind of ready to not do that anymore. So I got interested in the outdoors, and a friend of mine had like one of those little Nalgene hoses on his backpack. And I was like, oh, I want to know where do you get one of those? And I just got kind of interested in learning how to be outdoors. And that led to seeing a lot more things outdoors. And I don't think it was, I don't think I knew for a very long time that that's where I was going to stay. But I just kind of kept doing jobs that felt like me when I was doing them, and they never felt like desk jobs. So, and to speak to the women in science part, I think it's gotten a lot better over time. At least in ecology, it's gotten a lot better, but there are still really significant barriers. And a lot of it has to do with not being able to see yourself in the older generation, because there's what we call the leaky pipeline, where you have a lot of young female graduate students, and then when you get to the top positions, it's all white men who sort of make it through the sieve. And that's not because young women are not talented, it's because it's not necessarily a friendly environment in a lot of little ways.
Yeah,
And
I
I
have
would
a
say that that's,
PhD in physiology, and yeah,
yeah. And it's so amplified too for people of color. I mean, as a white woman, I've had so many privileges that people of color just do not have. And yeah, so you know how it is. And I would just say that you know, if you are a young woman and you think you might want to do it, those barriers don't make it impossible. And often the barriers that I've had have made it even more gratifying now that I'm sort of on the other side, because I get to mentor people who were like me or who didn't have the same opportunities even that I had. And that is so healing and so nurturing. And so it was worth the struggle, I would say. But my advice would be to find a good mentor who you feel like not only reflects you professionally, but also personally and who you'd like to be, and hold on to them for dear life.
Yeah, no, that's great advice. And we try to offer that with we have graduate students mentor, students in middle school and high school, and try to really like help them make that connection, make them see that like you don't already have to know that you want to be a scientist or feel that inside you somehow to end up there. We try to foster that curiosity in science, teach them how to, see things outside and observe and, ask questions and discover things. And I think it's really important to get people in science that are creative and curious. And so that's a huge part of our mission. So thank you for bringing your story. I just think it's really important, for our audience to hear that side and the stories of how we got there. So yeah, thank you everybody for joining tonight. And there's still time to donate. Big Day of Giving goes until midnight tonight. And our board is matching. So I really appreciate it. And we wouldn't be here without scientists like you and graduate students who help. And so thank you so much for joining us. We appreciate your time.
Thank you so much for having me and asking me to do this. And I do hope you all donate. What a wonderful organization. Thank you.
Thank you. Good night.
Good night.